Hello, my name is Dr. Paul Cottrell, and I'm doing this recording on October 21st. These series of videos that I'll be doing will be bite-sized concepts of a larger series that I did that was titled Biological Weapons in Foreign Policy. It was a three-part series. So please take a look at those videos. They were released maybe a month or two ago, but these videos that I'm going to be doing will be the bite-sized versions of those, trying to explain the importance of understanding biological weapons and foreign policy on all of its important facets and features. The problems that are set in offensive weaponry, the problems with defensive weaponry. So let's begin. The title of this video is Rapid Biological Discovery. All right. It's the overall concept is the challenges of the bio AI age. Okay. So what are some of the challenges in an age of biological discovery in artificial intelligence? So we're going to begin in this video about the rapid biological discovery. So with rapid biological discovery, let's take it in the context of just scientific endeavor and not biological weapons at this moment in time. Biological discovery is advanced to a very rapid pace. Why? We have means of looking through microscopes and electron microscopes, understanding the biochemistry of a system, what's going on in the cell. We have genetic manipulations of certain cell lines or either pathogens, and understanding how they can be, what some of those processes are. There's fluorescent proteins that can be attached, tag proteins that could allow a researcher to understand the flow of a particular process in a cell or in a tissue, understanding the affinities of certain types of molecules or certain types of proteins to another protein, how they work as agonists or antagonists. So there's a lot of discovery that can happen in a scientific endeavor through biotechnologies. All right. One classic example of biotechnologies is the ability to silence certain genes in a cell to understand how that cell's working or that tissue's working, or even at the animal level where you're doing a knockout of particular genes in an animal. Those are usually used in cancer studies, but you can understand how certain genes work by knocking them out. How do you knock out a particular cell in a cell line, or a particular gene in a cell line? Well, you can take the standard cell line and you can, through CRISPR, cut out a particular gene and grow those cells and filter out those cells that have had that gene removed, purify that culture, and then do your experiment, right? So there's means of inserting and cutting out certain types of areas of the gene. Another method is not just the CRISPR method where you can cut and insert, but you can also do restrictive enzymes that are looking for a particular target, and they can cut a certain region of the genome. All right. Another way to do it is in terms of silencing. If you know, let's say, the RNA of something, then you, in the cytoplasm, you can have that cell be exposed to the opposite, a complementary sequence, to silence that RNA that's going to produce a protein. Well, if you have a complement that binds to it, it can't. It can't create that protein. So there's different ways to silence genes, either permanently because of a knockout, or through other means like a complementary sequence that attaches. There's other means too where you augment and accelerate the process of breaking down certain types of proteins. So that if you increase the proteases, or if you create some sort of chemical that acts as an antagonist to a process, there's lots of ways to silence the expression of genes, or to do knockouts, or to do something upstream or downstream, depending on what you want to do. So in the scientific endeavor, the point here is that there's lots of means and tools to be able to manipulate cell cultures, even organs, organ systems, or organisms. You can breed certain types of animals to have certain features that are knocked out or expressed. So we talked mostly about knocking out certain types of genes, but you can also insert genes. You can insert genes at the genomic level through CRISPR or through recombinant engineering, where, instead of CRISPR, you may have a plasmid that comes in, and that plasmid is in an environment for that cell culture that allow for that plasmid to integrate into the genome. And then if it's in an environment where it's coded, then that gene will be expressed in that cell line. And you can test to see if that particular protein is being coded for. So the tools, the specificity, and the productivity of the biological tools have really advanced in the last 25 years or so, especially with CRISPR. All right. So you can basically, if you can dream up the experiment, you can basically make it. We're at that point in time now. Now, in terms of weaponry, you can use these types of systems in the biotech world, these tools, and do nefarious things. You can insert certain types of genes that may cause harm. You can have certain genes taken out to cause harm. You can manipulate pathogens. You can manipulate, certain types of therapies, if you really want to become nefarious, right? There are different things that can happen where you're building weapon systems, but you can also build defense systems too. So if you know what your adversary may drop on you in terms of a pathogen, and you understand what that genome is and what the mechanisms are that cause the pathologies, then you can mitigate it through therapeutics, through inoculations. You'll be able to target it better, right? If you know, let's say, the sequence for a higher affinity protein that's causing harm, and that your adversary created it, then you may want to purify that protein and create an inoculation, or isolate the code, insert it in a vector of some sort, and then allow that to be part of a biodefense system. So there's different types of biodefense. You have inoculations, you may have antimicrobials, you may have Other therapeutics, monoclonal antibodies. There may be a lot of different things that could be deployed as a biodefense against some sort of weapon that was used on your population. In addition, when you're creating these offensive weapons, you need to have an antidote, some sort of therapeutic, monoclonal antibody, antimicrobials, or you have an inoculation, whatever it is, to prevent your population from getting it when you deploy it. This is one of those things about bioweapons that one needs to think about is that there is a boomerang effect that takes place. There's a feedback loop in society. Once you release it, you can't really control it. It can't spread around. So if you can't contain the spread, then your population may be harmed. And the thing is that bioweapons and their biodefense for biological weapons, there's porosity. There's porosity in the bio shield. So you may reduce severity, but you probably can't prevent 100% the collateral damage to your society. So in the terms of rapid biological discovery with artificial intelligence, now that's the world that we're living in. And now you add in a layer of artificial intelligence. Now, why is this important? Artificial intelligence is going to be able to add to discovery, scientific discovery, and also weapon development and defense, offensive weapons and defensive biological weapons. So in the scientific category, if we want to look at it from that point of view, using AI for biological discovery, one way is to have an AI agent knowing all these different chemicals and therapeutics that are known, what they attach to, train the model that way, and come up with maybe novel uses of an already created therapeutic for certain diseases. That's one way to do it. Another way is to understand what the target region is for a particular chemical, acting as an agonist or an antagonist for a certain receptor or certain protein in the cytoplasm of a cell. And the AI knows the topology of that protein that you want to inhibit. Well, it may start with a base molecule, protein, or just some sort of chemical molecule, and try to find ways to add certain groups, organic groups, to the molecule for it to fit better in that shape, and then produce a result. And then it sends that data to a chemist, and the chemist will create it and then test it and validate. That's another way where in science, AI can be used to help. Because a lot of times what happens is that the chemist will go, okay, we have a basic idea what the shape is. And we think maybe if we do these different types of chemical shapes and rotations, that maybe it will inhibit, and they do maybe 100 different varieties of these shapes and then try each one. Well, you can speed that up in a simulation using AI. That's one way to do it. Another great thing about AI in terms of science is that you can take a shape and start from scratch or a base protein, and it will morph the protein to a point where it gets the best shape. All right, so it's doing three-dimensional shape and then testing attachment and pull to the target molecule, either acting as an agonist or an antagonist, depending on what you want to do. And it'll have maybe the top 5 or top 10 best shapes by just mutating the sequence, because a protein is a sequence of RNA. Well, if you're changing the RNA to get the best shape by running a simulation, right? The AI will give you maybe the top 5, right? So, but in reality, sometimes folding proteins is different than, let's say, in silico. So you have to validate it. So once you know the actual sequence, RNA sequence for that protein that has the best shape, or maybe the best 5 shapes, then you want to validate that in a biological experiment. So if it's a cell that's expressing a certain protein and you want to inhibit that protein, then what you want to do is have that protein express that this inhibitor expressed in the cell. So you infect the cell, have that cell express the inhibitor. That protein will be folded, right, in a certain way. You're hoping that that fold will be the same in the real biological system versus the in silico in the simulation. And you test to see if that inhibitor works. Some sort of cellular process stops. Maybe you want to try to fight cancer, or maybe you are trying to shut down a certain pathway. But you can do these experiments. So you can speed this up through AI. Another thing that AI can do in terms of weapon development and weapon defense is that if, let's say in the terms of weapon defense, if you have a pathogen that comes in from an enemy and is attacking your society, you can isolate it. So you have these isolates. You are doing a rapid next-gen sequencing because you don't know what it is, right? And you get the genome, right, through this next-gen sequencing. Once you have that genome and then you compare it to the database, you may say, this is a mutation of this type of pathogen, or it may be a brand new pathogen. And we'll discuss that in this video. So you have a rapid sequence. You know, let's say, the mutation of that base pathogen, the wild type. And by comparing the differences, you can make the assumption that those differences are what's making it more pathogenic. And so you target those particular changes in its genome. Usually it's some sort of attachment, some sort of spike, some sort of glycoprotein that's attaching to some sort of receptor. It doesn't always have to be that way. In some pathogens, it's just a protein that's expressed in the cytoplasm, and it's not about the attachment. It's normal attachment, but more it has an expression of other proteins that shut down processes and cause harm. So once you figure what's really happening, you can create, that gene from the weapon that is different, and you can create, using AI, you can say, okay, that gene is targeting this type of protein, or this type of receptor, or this type of pathway. So you want to do is just like go, okay, we can create therapeutics. There's lots of different things that we can create. We can create monoclonal antibodies. We can create antimicrobials or some sort of antivirals or whatever it is, whatever the anti-pathogenic therapy. We can do inoculations. So in terms of, let's say, typical therapeutics, you may want to block the receptor that is trying to attach to. All right. That could be off the shelf, or it could be brand new. You can do a monoclonal antibody where you're creating an antibody against the actual pathogen, either maybe the surface of the pathogen and attack its spikes, or you may want to create a inoculation or a monoclonal antibody against one of the proteins that it's creating in the cytoplasm. And so when it's expressed, your body already has the antibodies to come in and to attack. But probably the most effective way is to have a monoclonal antibody, at least at the early stage of the pathogen being released and attacking a certain population, is probably to have a monoclonal antibody against the surface of that pathogen. All right, but it depends on what you're talking about, because there are nuances where it may seem very benign on the surface, a pathogen on the surface, very benign, but it can be very problematic in terms of pathology with internal proteins that are being expressed in the cytoplasm. So there's different ways to do it. You want to elicit some sort of immune response, right? So another biodefense is to create antibodies against certain proteins, either the surface proteins or internal proteins of this pathogen that is causing harm. And you will have the antibodies to fight the infection, creating these antimicrobials. So the whole idea here is that, okay, if you know what it's attaching to, either a receptor, outside receptor with a spike, or some sort of internal process, you can stop it. You can block the receptor, or you can have antibodies that are against the pathogen, internal structure, internal proteins, and or surface proteins. In addition, a lot of times when it comes to especially viral, you want to have not a monotherapy, you want to have a polytherapy, meaning that you have multiple mechanisms being attacked to fight the pathogen. Maybe the entry to the cell, you block that. As the gene is being expressed to spread more viron, you may want to block that protein, right? So if you use a polytherapy instead of a monotherapy, usually it's like a tri-therapy. Different antivirals attacking different mechanisms on top of some sort of monoclonal or some sort of inoculation to elicit an immune response. When you have a society that has all these different tools to fight it, it's better, all right, instead of just one method. Because if it's only one method, then there is a selection, natural selection for mutations for that pathogen to circumvent the monotherapy. All right, so in terms of AI, you can, all right, once you know what that pathway you want to block is, let's just say it's an attachment to the cell surface of something, and you know the 3D of that receptor, and you know the 3D shape of the glycoprotein, then what you do is like, then you can go, okay, I can create, the AI can say, okay, I want to block some of these receptors. Now, if you block the receptors too effectively, it may cause harm to the body, even though it's blocking the pathogen, because these receptors are there for a reason, right? So if you're blocking a natural pathway for good health, then obviously it's going to cause harm. So that may not be the best way to do it. So another way is to create, if you know the glycoprotein shape, then you create a a protein that attaches. And so you inject that protein in the bloodstream or through some other means in the body. And what will happen is that that inhibitor will attach to the spike and act like a monoclonal antibody. So it's not a monoclonal antibody. It's actually a AI created code of figuring out what the RNA is for a particular shape for a protein to inhibit the spike. That's one way to do it. Another way is to know that that's what that code for the spike is through the sequencing, and then inject that protein, right? So you create lots of these proteins outside in vivo, and then you inject them into an animal model that can generate antibodies, and those antibodies can be used as monoclonals in the human. Another way to do it is to know the sequence of the protein, the glycoprotein spike, and then elicit a response to that protein through either an attenuated virus vector or an attenuated pathogen vector. Some are live. Some vaccines are live, inoculations are live. Others are, this lipid nanoparticle type. Others use different vectors, like an adenovirus vector or other vaccine vectors. So the point here is just that there are lots of ways to create an elicitation of the immune system to fight that glycoprotein from that pathogen that was the weapon. All right, so you have therapy, different types of therapies. You have the monoclonals, right? You have the inhibitors that are not monoclonals, but they're kind of act like a monoclonal. Then you have antivirals or antimicrobials, right? And you have other types of vaccines that could elicit an immune response against that glycoprotein. But where AI can come in here is knowing that shape, that protein shape, and taking a base protein and modifying that rapidly to get the best attachment, the best affinity, and the best pull. So it has the highest attachment and pull rating. And when you do that, you're blocking it. And once you know that, you know the code, you know the RNA code. And once you know the RNA code, then if it's going to be a therapy that's kind of like a monoclonal, but it's not. It's more like a protein. It's an injected protein. Then what you have is that you know what that code is. You make that protein in batches, typically usually it's in E. coli, but you're creating these batches, and then you are purifying it, and then you inject the protein and it inhibits it. So it's acting like a monoclonal. So that's where the AI comes in, is it's created the shape, knows the code for the RNA for that shape, and then you're creating them in the E. coli. But before you use this as a therapy, you have to test it to make sure that what the shape that the AI predicted is what really happens in nature. Because sometimes it's different, depending on the complexity of the shape. All right? Because you can have the right code, but slightly different environmental parameters, pH, salinity, different environments, temperature, whatever it is, different concentrations of different chemicals that can slightly change the shape. And it may be more effective, may be less effective than what was predicted in the AI model. Now, some AI models are working with different parameters, different pHs, different salt concentrations, different temperatures, and just to have a larger library of potential shapes to investigate. All right. So verifying it before usage is super important. But once it's verified, let's say it does work, then you're creating this mass batch of this protein as a therapy to block the pathogen. So that's that's a therapy. That's a therapeutic. But it acts kind of like a monoclonal, but it's not a monoclonal. Now, there are ways to manipulate cells that produce antibodies, B cells. You can manipulate them in a way where it can be producing antibodies. So in vivo. So if your AI knows what the antibody shape should be for blocking the protein structure of the pathogen, the glycoprotein spike, if it knows that shape, or a series of maybe 5 potential shapes, then you can manipulate the B cells to actually produce those antibodies in vivo. I'm sorry, in vitro, not in vivo, in vitro. And so when you do that, then you can purify, and then those antibodies that are being produced by a certain B cell line are monoclonals. And then you inject those monoclonals. And so it acts similar to the first example, where it's a protein that just inhibits the glycoprotein from the pathogen. So you're using AI to find the shape, and a antibody shape that can be used through a manipulation of B cells outside the body, create monoclonals, and then inject. Now, the typical way to do monoclonals is to use an animal model that's producing them, and then you purify and then you inject. But there's other ways to make monoclonals. So AI can help with that, right? Now, AI may also help with pathogens that have a glycoprotein, and the AI is running a simulation that what's the probability of certain types of mutations to take place in 10 generations of mutations or 100 generations of mutations. And there is some sort of probability cone that's created, that certain mutations in binding regions are more probable than others. Then you can not only have an inoculation sequence for the current glycoprotein, but you can have a series of inoculations on the shelf, codes for inoculations on the shelf, that will allow you to be ready for the potentiality of a certain mutation down the road. Now, what's the beauty of the immune system is there's a lot of cross-reactivity. So if an individual did not have an inoculation and didn't get the first variant of a pathogen, but contracted it 2 or 3 years later and it's mutation, and it happens to fall in that probability cone that was predicted by the AI, and they could use an off-the-shelf vaccine or inoculation or monoclonal or whatever that could inhibit that new mutated version instead of using the old inoculation. So there's a lot of ways to develop biodefense using AI. All right. Prediction of mutation of the pathogen, but also prediction of shapes that are needed to inhibit a certain protein on or in or produced by the pathogen. All right? Now, you can also use AI through rapid development for offensive means. So we spent some time on how to build a conceptual model of a defense using AI in biotech. Now we're going to pivot to offensive capability. So if you have a pathogen that you want to modify to use as a weapon, then let's just say we're using the glycoprotein example of some certain pathogen, maybe a virus. It doesn't have to be, but it could be, all right? And you have the AI know, the AI knows what receptors that usually attaches to, because there's a body of knowledge of certain pathogens that attaches to this receptor and this receptor and that receptor, right? So the AI knows what the different receptors are. And it's modifying, instead of a shape in the biodefense example, it was modifying a shape to block. What you're doing now is modify a shape on the glycoprotein to have a better affinity to a receptor. Now, in the olden days, you had to run multiple experiments and create mutations. There's different ways to create mutations, but you create mutations in a laboratory and you wait for a better affinity. Or maybe you do recombinant engineering and you add some ultraviolet radiation in certain lines and you reiterate this. Eventually, over time, you will have a pathogen that attaches to the receptor more efficiently. That's what Dr. Shi did in 2008. So for the crisis of 2020. So in the olden days, it was recombinant engineering and an iterative process to allow for a mutation to take hold. Then you select for better affinity and then continue that process, serial passage. And you keep on improving it, improving it. It takes time. Well, AI can do the serial passage much more rapidly. So what you're doing is you're taking the glycoprotein, in this case, and you're modifying it to a point where it's actually higher affinity to a receptor and a better pull. So it doesn't pull out. So it can attach. So you can have these, you can have it attach, but it pulls out easily. Right? So so you want it attaching, but also once it's attached, it doesn't pull out. Because once it's attached, then it can fuse into the cell. Right? So with this, you're telling the AI, modify the spike on the pathogen to have the best affinity. Then it knows the code. Then the AI knows the code. Once it knows the code, then it's simple biotech, where you're just inserting that new sequence that the AI predicted, put it into the pathogen, and then test in vitro. Test in vitro what your affinity is, right? And then once you know that, and then you move towards in vivo animal models that are representative of humans. And if that proves out to be correct, then now you have a weapon. And since you know the spike sequence, you can start creating the therapies against it. You know, these proteins that can inhibit these monoclonals and other inoculations through different vectors. Right? So there's lots of ways to use AI for defensive and for offensive capability in biological warfare. And it's all due to the rapid development in discovery. In an AI world, you can rapidly do this. Pre-AI, it was serial passage. Pre-AI, it was sometimes accidental. Pre-AI was a lot of doing experiments, doing, restrictive enzyme, recombinant methods. Sometimes it would work, sometimes it wouldn't. Then you start using CRISPR where it's a little bit more targeted, but you get out of target areas where you're cutting and pasting in a region that you didn't want. So the specificity is getting better for CRISPR, but it's not perfect. There's off target insertions and deletions. So, but the technology is much faster now. It's much better. Even without AI, biotech is much more effective and quicker. You add in the layer of AI, and then you're speeding up the discovery of what the sequence should be to have the best affinity, or what's the sequence to be to have the best antagonist, what's the sequence to be to build a pathogen that's the most lethal. But you can also use AI to figure out what's the best inhibitor to the pathogen. So you can use it as a shield for your own offensive biological weapons, but you can also use it as a shield against the potential use of biological weapons from your enemy. So it's really important to keep this in mind, that you have this, that there are these capabilities in biological warfare due to AI, and there's this rapid development that's taking place. And it has a lot to do with the ability for AI to compute quickly. It has a much larger knowledge base of different affinities, chemicals that can block or attach. It could, through generative AI, add certain groups for a chemical, delete or add certain sequences for a protein to have the better affinity. It can all figure this out. Now, could mankind do it without AI? Yes, but it's much longer. It takes much more effort to do. It takes much more time to do. But AI, it speeds it all up. So when you have this speeding up of biotech and rapid AI computation, then what you are having is a means to escalate the arms race in biological warfare. And this is why it's so important to do these videos and talk about how biological weapons are important to understand in terms of when to use them, when not to use them, the ethics of it, and the barriers in entry are very low. So you may have different types of actors. You may have state actors, you may have terrorist actors, you may even have lone wolves. And we're at the point, it's already happened, but we're at the point that you can take a certain type of pathogen and insert that genome into another pathogen, so that pathogen acts as a Trojan horse, and then eventually codes for the more lethal pathogen. So something that's relatively benign could have an insert that has a pathogen that is the code for that pathogen that is deadly, and it acts as a Trojan horse. This has already been developed. This is not theory. This is not scientific. This is not sci-fi. It's already been done. So we're already there. The barriers of entry are low, and you have different actors. Rational actors, where you would say that the state, at the state level or the country level, and then you have more irrational actors, terrorists and lone wolves. Now, how is it different than nuclear weapons and in foreign policy? Nuclear weapons, the barriers of entry are very high. In biological weapons, the barriers of entry are very low. So this is the reason why I'm doing these videos. So hopefully you understand how rapid biological discovery and rapid AI computation work together in terms of building biological weapons in an offensive capability and a defensive capability. That's the end of the video. Please help support the work that I do by making sure you go to my website and take a look at the products that I offer. If you like these videos and you want to support, get those products. And please donate. You can donate through Stripe, PayPal, or buy me a coffee on my website. You can also be a paid member to my Patreon channel and my Substack channel. I do all these videos for free. Thank you for listening and have a nice day.